This is our last lecture on chapter 24, and I want to pick up on that theme, the key concept of structure and function. If you recall, carbohydrates, linear carbohydrates such as cellulose, these are glucoses, monomers of sugar that are linked together to form these long chains, and they're straight, they're linear, and they sit flat on top of one another. They form these large strings, and that provides strength for the cell wall of plants. Now, if you have a branched carbohydrate, such as starch, this can't lay down flat, and so it has a different purpose, and it is a functional purpose rather than a structural one. Glycogen in animals is the same way, such as ourselves. We take the starch, it breaks down into monomers of glucose in our body, we reassemble that into these highly branched molecules of glycogen, and they are not able to lay down flat. They don't have a structural purpose, they have a functional purpose, and again, that's to store energy. We then looked at the lipids, such as the straight-chained lipids, such as the saturated triglycerides, and those are fats because the chains were full of hydrogens and they were all flat. The bent ones are found in plants, and those are the oils. That's because it has a double bond and they can't lay down flat. We then looked at the steroids as well, and so we're going to continue this theme of proteins, where we have a linear protein being more of a structural molecule, such as those that are found in our muscles. And then we have these functional proteins that are going to be folding up into globular shapes. And that's what we're looking at today. And that is going to be enzymatic activity, our enzymes. So our enzymes, they build things up and they break them down. A lot of people think that enzymes just cut things up into smaller pieces. Well, they do. If you remember the enzyme in your saliva or your spit, that cut up larger molecules of starch into glucose. So enzymes do break things down, but they also build things up. You might have remembered a molecule from biology, DNA polymerase, or even RNA polymerase. These are also enzymes, but they build molecules up. So enzymes can break things down, but they can also build up. What's the function of enzymes? So if you remember from our previous lesson, if you have a different amino acid sequence, we're going to have different R groups. Those are the side chains. And if you have different R groups, those proteins are going to fold differently. They can either fold into a linear shape or they can fold into a globular shape. So the different shape determines that different function. Each enzyme or protein has a different shape, means that each enzyme does different work. Now imagine if you take your car key and try to open up your house with the front door and the lock. It's not going to work, and vice versa. If you take your house key, that's not going to open up your car. Question is why? Well, each key has a different shape to it, and there's a different shaped locking mechanism on the inside of each of the handles of the door of your house and your car. So you have to have a matching molecule that matches the locking mechanism. And this is what we find on okay, here we have a picture. There is an enzyme. The enzyme is that purple blob. I'm going to highlight a couple regions here so you can see them as well. You'll see these openings here and here. These are called the active sites. They have those openings because the enzyme folded to that specific shape because of the sequence of amino acids. So the amino acids fold in a way that they can act on very specific molecules. So each enzyme shape will have a specific active site. This is the opening on the protein where the work is being done. Next in the picture, what you can see is this molecule over here, which is going to be sucrose. Even sucrose, which is a dimer of saccharides, it's a disaccharide, this can be broken down into individual monomers of glucose and fructose. This has the perfect shape to fit inside here. So the sucrose is the key, and the enzyme is like the door. It's the locking mechanism. So the shape of the active site accepts the specific starting materials. These starting materials are called substrates. So it's like a chemical reaction where we have our reactants, but when we're dealing with biological molecules, they're called substrates. Notice how in the next picture, what we see is we have the substrates, which is the sucrose, fitting perfectly inside the enzyme. And that's because it has the same shape that the enzyme has. The substrates enter the active site and the enzymes rearrange the bonds on the substrate, and a new molecule is released, or new molecules are released. So if you notice right here, we are going to be breaking that bond between those two, and we release the two products. That's breaking things down. You can also imagine if you're going in the reverse, you have two individual products that come in. Those are going to be bound to each other. They're going to be forming a bond, and they'll be released as one product. So here's another series of pictures showing you. So here's our substrate and the active site. So notice how it has a nice similar shape right here. We have this shape that goes, let me try to get a color. I don't know if I can highlight this. I'm unable to highlight this right here. But you can see that here's the shape on the enzyme, and it matches the shape on the protein, which is right here. Those are going to fit into each other. So this is when the two are bound together. This is when the bonds are rearranged, and it's going to release the two end products. How do these molecules do this? Well, if you remember from biology, there's this molecule, an energetic molecule called ATP. Enzymes don't really act by themselves. They need to be activated. This is going to a concept of you can't get something for nothing. Okay, are you going to go to your job and continue going to your job and not get paid? That might not make a lot of sense unless you're in volunteer work. However, with this ATP is going to be activating and giving energy. It's going to be giving payment to the enzyme so that the enzyme can do its work. So it's the same thing with the enzyme. It's not going to do work all by itself. It needs to first be encouraged by some ATP. So ATP, if you remember from biology, this is the currency of the cell. This is the provider of energy for your enzymes to do work. And energy is stored in the phosphate bonds of ATP, and it's transferred to other molecules in a process called phosphorylation. This transfers one of the phosphates. So we'll take a look at this after you record that information about transferring one of the phosphates. So ATP is called ATP because we're using a base which is very similar to the adenine that you find on DNA, and it's called adenosine. And to that adenosine, which is similar to the DNA base, if you remember your A's, your T's, your G's, and your C's, are three phosphate molecules, 1, 2, 3. Since we have three phosphates, the 3 is the tri. So we have adenosine triphosphate. Now, if you notice, I'm going to highlight these bonds that are between them. The bonds between the second and third are high energy bonds. They contain a lot of energy within them. And so what happens is this ATP over on the left is going to go to an enzyme, and it's going to transfer one of the phosphoruses with the high energy bond located right here, and that's going to give it to an enzyme. This is the payment. Remember, the ATP is the currency of the cell. It's going to be using its currency to pay other molecules to do their work. Since we're transferring a phosphorus, this is the process called phosphorylation. Notice what we end up with. Since we have now removed one of the phosphates from the ATP, we now only have 1 and 2 phosphates left. So we have 2 is the prefix for di, so we have adenosine diphosphate, or now we have our ADP. So the ATP is broken down into the ADP and the extra phosphorus, and it's that phosphorus in the process of phosphorylation that energizes the molecule. It's not just used for enzymes, but it's used for a lot of other processes as well. So phosphorylated molecules use the energy for work, and then it releases the phosphate after it's done. Take a look at the top picture. Here's a muscle fiber, looking at this one up here. Okay, this is a muscle fiber, and it's using the ATP. Okay, it's taking the ATP right here, and the ATP then activates this muscle fiber. It's sort of like a mousetrap. Okay, a mousetrap springs, of course, but you first have to add energy to the mousetrap by pulling the spring back. That's what ATP does to your muscles. It pulls your muscles and it gets them ready to snap and to move. And when triggered by the brain, then your muscle will then contract. And after it contracts, it's going to release the ADP and the P. So it's going to release the phosphate. So we now have released that. In the second picture, what we see is we have a transport protein. There are many proteins that are in your membranes of your cells, and these proteins are passive, meaning that the molecules simply flow through. However, the cell requires large concentrations of glucose, for example, and we need to pack glucose into the cells. Now imagine there's some type of dance club and you have a couple of bouncers at the front door, okay? And those bouncers have been paid to literally snatch people off the street and to push them into the dance club because they want as many people as possible in the dance club. So again, here, the bouncers are sort of like the enzymes that are in a cell wall. They take their payment and then they do work. So the bouncers were shoving people into the dance club, and enzymes do the same thing. They get phosphorylated by the ATP, and then what they do is they take a molecule that's in low concentration, that's where the street is, and they pack it into the cell where it's already high concentration. You know, things don't normally go from low concentration to high concentration, and that's why the enzyme or the protein that's located in the cell membrane needs to be paid to push the molecule to a higher concentration. In the last case, what we have is we have right here, we're making a molecule. We have a molecule, this molecule X has been phosphorylated and it needs to link to Y. And so we link those two together and it's going to be releasing the phosphate. So in all three cases, some activity is taking place. We're making a bond, we're transporting a protein, we're moving a protein, and all this was done with the help of ATP. So now we have all this ADP at the end, plus this phosphorus. We're going to recreate ATP because we only have about a 10-second supply of ATP. This is produced when carbon-carbon bonds, such as found in sugar, are broken. And this is what's called cellular respiration. So we have to produce ATP. Our bodies only have about a 10 seconds of ATP supply. If we run out of ATP, our body will cease to function and our cells will die. So we have to have readily access to ATP. And the way that we do this is after the ATP phosphorylates and does its work, okay, now we're in the form of ADP and the phosphate. We need to bring those back together, and that's done through cellular respiration. We take our sugars or other molecules, which we'll see in a few minutes, and we will break those down, and those carbon-carbon bonds are like the glow stick. We snap those bonds in the carbon-carbon bonds of sugar. The energy that's stored in those bonds is then used to bring back the ADP and the P together to make ATP. This is the whole key concept of what happens inside of our bodies. So how do we create the ATP? Well, if you remember photosynthesis and respiration, they're complete opposites of each other. So we're going to be looking at respiration, which is the opposite of photosynthesis. So if you remember, if you take sunlight and carbon dioxide and water, put those together in a plant, you'll be able to create glucose and oxygen. Well, now what we're doing is we're taking the glucose from the plant, either directly from sugars or from starch, and we're going to be using oxygen. We're going to burn those in the presence of oxygen like a candle is burning in order to produce energy. This is the process of aerobic respiration when we have enough oxygen present. So here's the formula, C6H12O6. Here's the glucose plus the oxygen produces carbon dioxide and water, and it releases ATP. So sugar is burned in oxygen, produces carbon dioxide, water. Now, you probably remember the Krebs cycle from biology. We're going to very briefly take a look at this process of producing energy. I'm going to give you a summary of the steps, and then there's going to be a drawing, and then after this drawing, we're going to summarize the entire chapter and see how every molecule that we looked at, whether it's going to be a protein, a fat, or a carbohydrate, can be used and broken down to release ATP in your body. So first of all, this is the Krebs cycle. This is taking place on the inside of your mitochondria. Remember the mitochondria? These are the powerhouses of the cell. This is where our energy is being produced. So glucose is first broken down into 2 3-carbon sugars. Okay, so we're first starting off with a 6-carbon sugar. Okay, I'm going to write that up here. So first we're starting off with a 6-carbon sugar, and then that's broken into, we don't need to know the name of it, but if you count these dots are the carbons. It's broken down into a 3-carbon sugar. Okay, that 3-carbon sugar then gets a carbon dioxide ripped off, and since we've had the carbon dioxide ripped off, this now has 2 carbons. So now we're at a 2-carbon sugar. Okay, this is slightly modified into this molecule you might remember from biology, acetyl coenzyme A. This 2-carbon molecule right here, this is what enters the Krebs cycle. Okay, so each 3-carbon sugar has a carbon dioxide removed. Each 2-carbon enters the Krebs cycle and produces carbon dioxide and energy to make ATP. So take a look at this process here. We're going to summarize this in the picture as well. We have a 2-carbon sugar coming in. That's the acetyl-CoA from the previous picture. We meet that up with this 4-carbon sugar. 4 carbons plus 2 carbons is equal to, you guessed it, 6 carbons. So you'll notice we now have a 6-carbon molecule right here. This is not glucose, but it's another molecule within the Krebs cycle. And then this is broken down into a 5-carbon, then back into a 4-carbon as well. So what I'd like to do is to end the video here, and I'd like you to go into the next video, and it's going to show you a brief representation of the Krebs cycle in slightly more detail.